have sensory receptors that are very much larger than what we've been talking about. They're more complex, and they are all found within the organ, within the head. Smell is the olfactory organs in the nasal cavity. Taste are the taste buds in the oral cavity. Hearing and equilibrium are both within our ears, and sight within our eyes. So synesthesia is a joint sensation, is a good way to describe it. It's a condition in which the brain interprets a stimulus for one sense as coming from another. Some people might say the paint smell blue. There are about 1 in 2,000 people with this condition. It lasts a lifetime. It's linked to creativity, has been discussed. It can be caused by a genetic mutation, and there are up to 4 genes that have been identified and linked to this synesthesia. Most common form is the grapheme color type, in which letters, numbers, or time evoke certain colors. We also have lexical gustatory sensation, when a name would evoke a strong taste or smell. Let's start with the sense of smell. The sense of smell is called olfaction, and the olfactory receptors are chemoreceptors. They respond to chemicals dissolved in liquids, and the liquid would be the fluid in your nose. Sense of smell provides about 75 to 80% of our taste. So we can smell things. Things can have a flavor, but a true taste comes from a combination of taste and odor. I'm sorry, true flavor and odor. The olfactory organs have receptors cells, which are bipolar neurons. They are supporting epithelial cells. The neurons have knobs at the distal end of their dendrites, and those are covered with cilia. They cover the upper parts of the nasal cavity and the superior nasal conchae, and then a portion of the nasal septum. It's estimated there are about 400 types of olfactory membrane receptors, and their depolarization their binding of those would result in combinations of depolarization and action potentials. The pathway for these signals, once the olfactory receptors are stimulated, goes from a nerve impulse in the cribriform plates to the ethmoid bone, where it runs into the olfactory nerves, which go to the olfactory bulb, the olfactory tracts, the limbic system for emotions, and the olfactory cortex for interpretation. The olfactory bulbs process sensory information, while the limbic system, the center for memory and emotion, provides an emotional response to certain odorant molecules, and sense of smell is one of the very powerful triggers for memory. Leading hypothesis for encoding those specific smells is that each olfactory receptor contains only one type of membrane protein. Each type of membrane protein will bind to several types of odorants, and the brain interprets this binding in what is called an olfactory code. So the olfactory organs are located high in the nasal cavity above the pathway of inhaled air. Their receptors undergo sensory adaptation quite rapidly. Your sense of smell can drop by 50% within 1 second after stimulation. Olfactory receptor neurons are the only ones in direct contact with the environment and can be damaged, but those receptors are replaced regularly, and it's very unusual for neurons to do so. Gustation is the sense of taste. Taste buds are the organs of taste. They're located on the papillae of the tongue, the roof of the mouth, the lining of the cheeks, and the walls of the pharynx. There are about 10,000 taste buds, and each has between 50 and 150 taste cells within them. The taste receptors are also chemoreceptors. Taste cells are modified epithelial cells functioning as receptors. They have cellular hair on them, or microvilli, that protrude from the taste cells through the pores of the taste buds to the sensitive parts of the taste cells. And these cells are also replaced regularly. Taste cells are replaced approximately every 3 days. There are 5 primary taste sensations: sweet, stimulated by carbohydrates; sour, by acid, that would be hydrogen ions; salty by metals such as sodium or other salts, positively charged salts. Bitter would be stimulated by a number of organic compounds as well as magnesium and calcium salts. Umami is stimulated by some amino acids. MSG is a primary target of this type of receptor. Each flavor represents one primary taste sensation or combination. Spicy foods might stimulate a class of pain receptors as well, and taste receptors also undergo rapid adaptation. So sensory impulses from the taste receptor travel along the fibers of 3 different cranial nerves according to the location of the taste cells. Could be facial, glossopharyngeal, or vagus nerves. Remember those cranial nerves. Cranial nerves conduct the impulses into the medulla oblongata, and they proceed to the thymus, where they are interpreted in the gustatory cortex, which is found in the insula region of the brain. There are a few smell and taste disorders. They can be caused by colds, flu, allergy, polyps, swollen mucous membranes. A head injury can do it, toxic chemical exposure, nutritional or metabolic problem or disease, and often the cause cannot be identified. And we also know that the COVID-19 SARS-CoV-2 virus has effect on smell and taste in a number of individuals. And also drugs and medications can alter smell and taste. Let's talk about hearing. The hearing is done in the ear. The ear is divided into 3 sections: the outer external, the middle, and the inner internal ear. The outer ear is part of our sense of hearing. It's divided into the auricle, which is a funnel shape that collects sound waves, more correctly called the pinna. The external acoustic meatus is a tube, an S-shaped tube, lined with ceremonous glands, carries sound to the tympanic membrane. When the sound waves reach the tympanic membrane vibrates. The tympanic membrane itself is the eardrum, which is again vibrating in response to sound waves. The middle ear would have the tympanic cavity, the auditory ossicles, and the oval window. The tympanic cavity is an air-filled space in the temporal bone. The ossicles are 3 bones. They vibrate in response to tympanic membrane vibrations. That serves to amplify. They're made up of the malleus, incus, and stapes, or as we sometimes refer to them, the hammer, the anvil, and the stirrup, so that when the tympanic membrane moves, it moves the malleus, which pulls on the incus and pushes on the incus, and then the stapes, which is attached to the oval window, that vibrates, moving the fluids within the inner ear where sound will be detected. So that there is also a tympanic reflex muscle. When you hit a loud sound, that muscle will contract to serve to dampen the receptors so that they don't move as much. There are the auditory tubes, formerly called the eustachian tubes, that will connect the middle ear to the throat. They help to maintain equal pressures on both sides of the tympanic membrane. They're usually closed by a valve-like flap in the throat. The inner ear is a complex system of labyrinths. There's the osseous labyrinth, the bony labyrinth, and the membranous labyrinth. The bony labyrinth is a canal in the temporal bone. It's filled with perilymph fluid, whereas the membranous labyrinth is a tube of similar shapes that lies within the osseous labyrinth, and these are filled with endolymph, a little different fluid than in the bony labyrinth. There are 3 parts to the labyrinth: the cochlear, the semicircular canals, and the vestibule. The cochlear function in hearing, semicircular canals in dynamic equilibrium, whereas the vestibule functions in static equilibrium. So equilibrium when we are moving for semicircular, when we're standing still for the vestibule. The cochlea are a small snail-shaped tube. It's widest at its base. It gets progressively narrower It's coiled around a bony core called the modilus. The spiral lamina is a bony shelf that coils around the cochlea. There are two windows in the wall of the bony labyrinth, the oval window and the round window. The oval window is an opening in the wall of the tympanic cavity, which the stapes transfers the vibrations to the fluid of the inner ear, and those vibrations will stimulate hearing receptors. The round window is a window in the wall of the inner ear that faces the tympanic cavity, through which excess vibrations dissipate into the tympanic cavity. The cochlea have three compartments: the scala vestibuli, the scala tympani, and the cochlear duct. The vestibuli are the upper compartment. They lead from the oval window to the apex of the spiral. They're part of the bony labyrinth. The scala are the lower compartment. They extend from the apex of the cochlea to the round window. They are also part of the bony labyrinth. And the cochlear ducts are in the middle compartment. They are portions of the membrane labyrinth in the cochlea itself. And they contain the vestibular membrane, the basilar membrane, and the tectoral membrane. Vestibular membrane is going to separate the scala vestibule from the cochlear duct. The basilar membrane is going to separate the cochlear duct from the scala tympani, and the tectorial membrane is going to extend partially into the cochlear duct. It's part of the hearing receptor organ, the spiral organ. Getting a cochlear implant is discussed in your book. You see a very nice image of a young boy who probably hearing something for one of the very few times he has in his life. It amplifies sound, but the hearing aids will amplify sound, but the cochlear implants stimulate the auditory nerve, and that will enable people to hear certain sounds. Before age 3, while the brain is learning to process speech and hearing, these tend to be most successful. So the spiral organ, formerly known as the organ of Corti, is the organ used for hearing. It sits on the upper surface of the basilar membrane, contains hearing receptor cells that are called hair cells. Those hair cells contain stereovilli or stereocilia. Along the tectorial membrane, they pass that acts like a roof over those hair cells, and sound vibrations will cause the stereocilia to contact and bend against the tectorial membrane. Different frequencies of vibration move different parts of that basilar membrane, and those different parts of the basilar membrane are interpreted as different frequencies when the nerve impulses are generated and interpreted in the brain. So the cochlear branch of the vestibular nerve goes to the medulla oblongata, to the midbrain, to the thalamus, and then to the auditory cortex in the temporal lobe of the cerebrum. Hearing loss. About 8% of people have some degree of hearing loss. There are two major types, conductive and sensorineural. In conductive deafness, interfering with the conduction of sound vibration to the inner ear, that accounts for approximately 95% of hearing loss cases. It may be due to an accumulation of earwax, hardening or injury to the tympanic membrane, injury to the auditory ossicles, or osteosclerosis, or otosclerosis, I'm sorry, not osteo. There are diagnostic tests to use, and the ones that would distinguish conductive deafness would be the Rhine and the Weber. In sensorineural deafness, there's damage to the cochlea or the auditory nerve or nerve pathways. That can be caused by long-term exposure to loud sounds, factory noise, loud music, or explosions. It can also be caused by other things that damage the central nervous system, such as tumors, strokes, certain drugs. So in order to get the impulse from the ear to the brain, first sound waves have to enter the external acoustic meatus. They have to cause the tympanic membrane to vibrate. The auditory bones, ossicles amplify that vibration, and the movement of the stapes against the oval window will transfer those vibrations to the perilymph. The vibrations through the vestibular membrane enter the endolymph of the cochlear duct. different frequencies will move specific regions. A receptor depolarizes its membrane, becomes more permeable to calcium ions, and the vesicles at the base would then release neurotransmitter, and neurotransmitter would stimulate nearby sensory neurons, and those sensory neurons would conduct along fibers of the cochlear branch to the vestibulo-ocular nerve, and then the auditory cortex would interpret those signals. Equilibrium is the feeling of equilibrium or balance. It's derived from two different senses or two different types of measurements working together. Static equilibrium, which senses our position when the body is not moving, that has receptors in the vestibule of the inner ear. And dynamic equilibrium, which senses the rotation and movement of our head and body. Their receptors are found in the semicircular canals. The euticle and saccule are expanded chambers of the membranous labyrinth and vestibule, and each contain maculae, which are little rocks. They are the organ of static equilibrium. It's a patch of hair and supporting cells, and they are embedded in a gelatinous material with the otoliths. Those are actually the rocks on their surface. Gravity will pull on that gelatinous mass, and the head changes positions. The otoliths will change position, pull on the gelatinous mass that's still in the hair, and the bending of those hairs will generate nerve impulses in the vestibular branch of the vestibulo-ocular nerve. The receptors for dynamic equilibrium are found in the three semicircular canals. Each of those canals is at a right angle to each other. Each canal contains an ampulla, which is a swelling of the membranous labyrinth that communicates with the vestibule. The crista ampullaris is a sensory organ for dynamic equilibrium. Here the hair cells and supporting cells are found. They're located in the ampulla within each semicircular canal, and they are hairs extending upwards into a dome-shaped gelatinous mass called the cupula. Rotation of our head, our body, is going to bend that cupula, stimulating the hair cells, and nerve impulses would then be sent all over the vestibular branch of the vestibular-ocular nerve. Our sense of sight. Visual receptors are found in the eyes. There are some accessory organs for the sense of sight. Upper and lower eyelids, eyelashes, lacrimal apparatus, and extrinsic eye muscles. Eyelids are called palpebrae. There are 4 layers. There's a skin, muscle, connective tissue, and conjunctiva. The orbicularis oris muscle closes the eyelid. The levator palpebris superioris muscle opens the eyelid. Tarsal glands secrete oil into the eyelashes. Now you hear tarsal, you might think foot immediately, but this is in the eye. Conjunctiva is a mucous membrane that lines the eyeball and covers the portion of the eyeball. The lacrimal apparatus would include the lacrimal gland, which is in the orbit lateral to the eyes, which secretes tears. Caniculi are 2 ducts that collect the tears. The lacrimal sac collects the tears from the caniculi, lines in a groove along the lacrimal bone. Collecting from the lacrimal sac is the lacrimal duct that empties the tears into the nasal cavity. One of the things in tears is an enzyme called lysozyme, which has antibacterial properties. The extrinsic eye muscles would be the superior rectus, the inferior rectus, the medial rectus, the lateral rectus, the superior oblique, and the inferior oblique. Rectus muscles rotate eyes in one direction, oblique at an angle. Eye itself is a hollow spherical organ of sight with 3 layers, an outer, middle, and inner, the fibrous, vascular, and nervous tunics. The outer fibrous tunic is cornea and sclera. The sclera is the anterior 1/6 approximately. It's the transparent part. It helps us to focus light, transmits and refracts light. The sclera is the other 5/6, more to the back. It's white, opaque, it protects the eye. It's attachment site for eye muscles, extrinsic eye muscles, and it's pierced by the optic nerve and blood vessels in the rear. The middle tunic, the vascular tunic, is choroid coat, ciliary body in the iris. The choroid coat is about the back 5/6. It provides blood supply, contains melanocytes, and the melanin they produce help to absorb extra light. Ciliary body's in the front. It's the thickest portion and pigmented. It forms a ring to hold the lens and changes the lens shape for focusing. The lens itself is anterior to the ciliary body in front of the lens. The iris is anterior to the ciliary body in front of the lens. It is pigmented and helps to control the amount of light entering the eye. The lens is a transparent biconvex behind the iris elastic held in place by suspensory ligaments used to help focus light rays and change the shape for long distance or close vision. Ciliary body forms the internal ring around the front of the eye, and its ciliary processes are the radiating folds. The ciliary muscles will contract and relax to move the lens. Suspensory ligament holds the lens in position, and the lens itself is right behind the iris and the pupil. Accommodation is a change of the shape of the lens to view close objects. The lens will become thicker, more convex when close objects, and it will also become flatter when focusing on distant objects. The ciliary muscles relax the suspensory ligament during accommodation. Again, the iris controls the amount of light entering the eye. It's got connective tissue and smooth muscle. Has the pupil is the window or opening in the center of the iris. Dim light would cause the stimulation of the radial muscles to dilate the pupil, whereas bright light would stimulate circular muscles to cause the pupils to constrict. And it is the amount and distribution of melanin that determines our eye color. A little interrupted there on this total video. This may be a little bit of a repeat, but I hope not too much anyway. So the aqueous humor is the fluid in the anterior cavity of the eye. It's secreted by the epithelium on the inner surface of the ciliary body. It provides nutrients and helps to maintain the shape of the anterior part of the eye, and it leaves through a canal called the scleral venous sinus, which you may see in some places listed as the canal of Schlemm. The inner tunic is the nerves. That's the retina. The retina has visual receptors, which are photoreceptors that are continuous with the optic nerve in the back of the eye. It ends just behind the margin of the ciliary body toward the front of the eye. It's composed of several layers. There's the macula lutea, which is a yellowish spot in the retina. The fovea centralis, which is the center of the macula lutea, which has the most cone receptors and produces our sharpest vision. The optic disc is more commonly referred to as our blind spot. It has no photoreceptors. It's found where the nerve fibers from the retina leave the eye and become the optic nerve. And the fluid in the back of the eye is called the vitreous humor. It's a thick gel that holds the retina flat against the choroid coat and helps maintain the eye's shape in that region. The posterior cavity is spaced and closed by the lens. The ciliary body in the retina has vitreous humor. That's a thick gel that supports the internal structures and maintains the shape of the eye again. The major groups of retinal neurons would be photoreceptor cells, bipolar cells, and ganglion cells. They provide the pathways for impulses that are triggered by photoreceptors to reach the optic nerve. There are also horizontal cells and amacrine cells which modify and integrate impulses. I won't go through the summary of the layer of eyes, but you, again, should use this table in your text to help you with that. So we will talk a little bit about the bending of light, which is called refraction. That occurs when light waves pass at an angle between mediums of different densities, and it's going to be involved in focusing so that the image falls for us on the fovea centralis. We can do that with a convex lens, which causes light waves to converge, or a concave lens, which causes light waves to diverge. As we focus in the retina, it's going to hit a convex surface on the cornea, a convex surface on the lens. The image is focused in the retina. It's a real image, which means it's upside down and reversed, and the visual cortex will correct that reversal for us. There are some disorders that can happen because of having an incorrect focal point. If the eye's too long, we have myopia. The image is focused before the retina and it's diverging when it reaches the retina. In a normal eye, the image is focused right on the retina. And in a hyperopia, the image has actually focused behind the lens, and the light rays are still converging. We use concave lens to correct nearsightedness and a convex lens to correct farsightedness. Photoreceptors are rods and cones. The rods are long, thin projections that have light-sensitive pigment called rhodopsin. They're 100 times more sensitive to light than cones, so they provide vision in dim light. They produce vision without color. It is in shades of gray. And they can only see rough outlines of an object. We don't get sharp edges to an object. Cones are short, blunt projections. They have light-sensitive pigments. They have 3 of them: erythrolab, kohlrabi, and psilorab. They provide vision in bright light and produce sharp images, and because each of those pigments absorbs different colors, by combining them we can get color vision, and the fovea centralis only contains cones. So rhodopsin is a visual purple. It's light-sensitive pigment in the rods. In the presence of light, it decomposes into opsin and retinol. That triggers a complex series of reaction that initiates a nerve impulses. Those impulses travel along the optic nerve. The iodopsins are the pigments in the cones. Each one contains a different light-sensitive pigment. Each type is sensitive to a different wavelength. The color perceived depend on which type of cones is stimulated: red, green, and blue. Stereo vision is because we get an image of the same object from 2 slightly different angles, and our brain puts that together to give us an idea of 3 dimensions. The visual pathways go through the ganglia cells to the retinol of the optic nerve, the optic chiasma, the optic tracts, the thalamus, the optic radiations, the visual cortex, which is found in the occipital lobe of the cerebrum. A few of the fibers branch off before reaching the thalamus, and they enter the nuclei used for visual reflexes. Over the course of our life, we get changes in our senses. We talked a little bit about age-related hearing loss. Age-related visual problems can include dry eye, floaters, loss of elasticity of the lens, decreasing accommodation, glaucoma, cataracts, macular degeneration. Smell and taste, usually due to the loss of olfactory receptors. And that wraps up Chapter 12, and I'm happy to say Semester 1. I'm looking forward to you coming back for Semester 2.